Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Achieving High-Quality, Efficient mRNA Synthesis for Streamlined and Cost-Effective Manufacturing. I'm Roisin McGuigan, Editor of Nucleic Acid Insights, and joining me today I have Helge Zieler and Khaled Yamut, who will explore how custom polymerases can enhance IVT efficiency, improving mRNA yield, quality, and purity for cost-effective manufacturing. After the presentations today, we'll have a live Q&A session. So feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. Now, I'd like to introduce our presenters. Helge, CEO of Primrose Bio, leverages decades of biotech experience to advance nucleic acid and protein manufacturing for genetic medicines, vaccines, and biotherapeutics. Khaled, CEO of Yamut Chem Consulting, is a leader in analytical sciences, quality, and manufacturing, with expertise in mRNA vaccines, regulatory support, and biopharmaceutical development. So without further ado, I'll now hand over to Helge to kick us off with the first presentation.
Thank you, Roisin. It's a great pleasure to be here today. My name is Helge Zieler. I'm CEO of Primrose Bio and really pleased to be able to tell you about our offerings in mRNA manufacturing. mRNA, as we all know, in the meantime, is one of the exciting new modalities in medicine. It came into everybody's view during COVID and clearly has developed into a new revolution in medicine as a genetic medicine that's based on genetic sequences delivered into the human body that encode proteins. And driven by the success of the COVID vaccines, this field has greatly expanded and is now being applied to vaccines against infectious diseases, cancer immunotherapy, cell and gene therapies, protein production, and a number of other ways of curing and preventing disease. Other potential areas being explored that won't be a topic today, but that we also keep an eye on, is potential applications in agriculture, animals, and plants. The mRNA space has greatly expanded since COVID, driven by the COVID vaccines. And so there are now around 200 companies and institutes active in this space, and they're developing hundreds of new mRNA-based vaccines and therapeutics. These span all the different indications that I just mentioned, and most of these are early stage. However, most importantly, a few mRNA-based vaccines have already been approved, and they have shown excellent efficacy, safety, and commercial success. So based on this expansion in the mRNA space, it's really a great pleasure for companies like Primrose Bio, and we are a supplier of manufacturing technologies for pharma to contribute to this exciting and expanding field. So Primrose, San Diego, California-based biotech company, offers manufacturing solutions for nucleic acids on one hand, and this is where our presentation today will focus, and therapeutic proteins on the other hand. And our nucleic acid offerings are centered on what we call the prima RNA pols, RNA polymerases with higher performance that allow more efficient, cost-effective manufacturing of higher purity, higher quality mRNA. And so to give you a bit of background on what this means, we have been focusing on this field for a number of years, and shown on the left, we've been developing RNA polymerases that can most efficiently produce mRNA for uses in pharma and in clinical materials. So that means that an RNA polymerase, which is the key enzyme for synthesizing mRNA from a DNA template, needs to produce RNA at high yields. It needs to produce high purity of RNA, high integrity, that is full length, and free of aborted transcripts, and RNA that's free of other contaminants that are undesirable, for example, double-stranded RNA. And then on top of that, there's a requirement for these RNAs to be capped. So the capping efficiency of the RNA polymerase using co-transcriptional capping needs to be able to accommodate the dominant cap of today, CleanCap AG, but also the increasing diversity of other cap analogs in development. And most importantly, when it comes to capping, the polymerase needs to be able to utilize the cap analogs efficiently, again, to lower cost of manufacturing. Finally, we're looking for enzymes, or we've been developing enzymes that can handle long templates. So-called self-amplifying RNAs tend to be as long as 10 or 15 kilobases, and that requires a high processivity enzyme that can go from start to finish without falling off. And then these enzymes have to have a high fidelity so that the template DNA is copied faithfully into mRNA without errors. All of these key performance indicators, or KPIs, are what we're working on simultaneously to offer improved and superior RNA polymerases to the industry. And what we're aiming at in terms of the process goals for manufacturing comes down to quality and cost. We are aiming for the in vitro transcription process, or IVT process, to have high yields and high capping efficiency with low inputs, low concentrations of the key expensive ingredients in the reaction. Also, we're aiming at high RNA purity in the reaction so that the cost of cleanup is reduced. And finally, as already mentioned, high yields and high quality RNA from long templates, and this will be a particular focus of today's presentation. So with these challenging requirements in mind, Primrose Bio brings a number of key capabilities that we've built up over the last few years that give us the ability to develop this class of enzyme and develop the best-in-kind RNA polymerases for mRNA manufacturing. Our work on RNA polymerases starts with a panel of RNA polymerases. We began our engagement in this space believing that one enzyme could not handle the full diversity of template lengths, template sequences, modification chemistry, cap analogs, cap chemistry, and other features and aspects of mRNA that make the space increasingly diverse. So we began with a panel, a collection of RNA polymerases, so that for any given chemistry or requirement, we would have a good starting enzyme. And then we developed a highly sophisticated molecular evolution enzyme to allow us to develop superior enzymes using very high throughput screening methods and the most modern synthetic biology tools to pull out and develop these improved enzymes along the qualities that I just listed. Third, out of this screening process and this molecular evolution process, we then isolate improved RNA polymerases. And these add to our collections of available enzymes that we can make available to customers that then meet all the different manufacturing needs of a customer and of the space of the industry. And finally, Primrose Bio has a proprietary protein production technology that we call the Phoenix expression technology that is able to, at low cost, produce very high purity enzymes. And so within this space of capabilities, we discover, develop, offer, and produce the best RNA polymerases for mRNA manufacturing. To illustrate in a schematic manner the improvement, the molecular evolution that we bring to the table, I want to show the four key performance indicators of the RNA polymerases in a schematic manner in this diamond-shaped diagram. So starting at the top clockwise, yield, integrity, double-strand RNA levels, and capping efficiency are each shown for a given enzyme. And yield, integrity, and capping efficiency need to be high, while double-stranded RNA needs to be low, very low, or absent. And so you can see on the diagram on the left, the dotted line represents the performance of the standard T7 RNA polymerase, which is still the state of the art in the industry. And T7 is a good enzyme, but not as good as it could be, and does have some flaws, as Khaled will point out a little bit later. And in comparison to the T7 performance, and here everything is sort of schematically shown in an illustrative manner with the numbers normalized to T7, you can see the performance of our prima RNA poles with, in general, higher yields, comparable integrity, lower double-stranded RNA, and comparable, although in this case, lower capping efficiency. And these are our first-generation enzymes. This is where we began several years ago. Now, after one or more rounds of molecular evolution, we can dramatically boost this performance. And our enzymes tend to have much higher yield, comparable or better integrity to T7, either very low or undetectable levels of double-stranded RNA, and very high capping efficiency across all available chemistries. So this, again, this is an illustration of enzyme performance. Khaled will show you very specific examples of specific enzymes we've developed. But in general, this is what we're able to do. This is proven that our system can make these types of advances and deliver polymerases with improved manufacturing qualities. And then it needs to be pointed out that some of the KPIs that I listed earlier, for example, cap incorporation efficiency, which impact costs by allowing the reduction of the cap analog concentration in the reaction, these are also addressed in this process of molecular evolution. What this means for the customer is briefly shown on this slide. So our value proposition is to lower mRNA production costs while either maintaining or increasing quality. And so again, schematically, breaking the cost down between reagents and consumables and fixed production costs, you can see the transition between T7 RNA polymerase on the left and our first generation primas as about a 20% reduction of production cost. And by the time our enzymes are highly evolved, and Khaled will show you some examples of these, we will achieve reductions of up to about 50%. So this is very substantial, and we're not stopping there. Our dedicated evolution and improvement machine will continue to turn and continue to produce improved enzymes. And so we see these performances dropping continually over time, but also most importantly, we will continue to be able to address all the diversity in chemistry, modifications, structure of RNA that the highly creative pharma industry and vaccine developers are coming up with. So with this, I'll hand over to Khaled to give you a sense of the performance of our polymerases.
Thank you, Helge, for the introduction. I'm Khaled Yamud. I'm the CEO of Yamud Chem Consulting, and I am currently also a strategic advisor for Primrose Bio. In the next slides, I'll be showing the importance of polymerase and how Primrose polymerases could become the standard for IVT reactions. So first is a brief introduction about the anatomy of mRNA and what raw material goes into the mRNA. There are currently three popular or emerging types of mRNA. You have the non-replicating mRNA, which is less than 10 kb, and that was explained by the COVID vaccine. And then you have the self-amplifying mRNA and circular mRNA. And the raw materials that goes in it all the three types of mRNA is pretty much kind of the same. You need your nucleotides, triphosphates. Those NTPs are the actual building blocks of the mRNA. You need your linearized plasmid to transcribe the mRNA, and overall, you need the engine that makes the mRNA, which is the RNA polymerase, and that's what we're going to focus on today. As for the synthesis of mRNA, you take a linearized DNA template, whether it's synthetic or from a DNA plasmid. You add your polymerase, your capping analog, and your NTPs, and you end up with multiple mRNAs there. You can have the full-length mRNA uncapped. What you're looking for is the capped mRNA, and also the truncated mRNA through aborted sequences. Now, the question becomes is all of that impacted by the polymerase performance in the IVT reaction. There's two ways to go about that. You can use a polymerase that give you all these impurities, and then you struggle downstream to purify them, or you eliminate all of that at the onset of the reaction to simplify and lower your cost of the downstream purification. So the goal here is to produce an ideal mRNA product that is over 90% full length, very high capping efficiency, very low to no double-stranded RNA, and also high fidelity copy of DNA template. So those are the goals that Primrose had in mind to achieve as they developing the polymerases for IVT reactions. So what I would like to also point out is, and Helgi pointed to that also, is not all polymerases are equal. You cannot expect one polymerase to perform the same regarding different capping strategies, sequences, any other strategies, circular, whether you're using native NTPs or modified NTPs. So all of that plays a role in the polymerases. And to understand that, one needs to understand a little bit about the polymerase mechanism in the IVT reaction. And so you design the polymerase with a promoter, with a specific promoter that binds to the template. So the polymerase goes in and search for that promoter. Once it's find the promoter, it'll bind to it and melt. And then it goes into the transcription initiation. At that point, where you have the transcription starting site, the polymerase will unwind, the DNA template will unwind, and the initiation of the transcription will start. From there, it goes into an abortive cycle, what we call an abortive cycle, where the polymerase could stall into this area and start producing truncated mRNA and abortive sequences. And then once it escapes that promoter area, it enters the elongation phase and start transcribing the full length of the mRNA and so forth. So if we look at the abortive cycle with the production of truncated mRNA, this is where your double strand will start to increase, because those truncated mRNAs have to go somewhere. And a lot of times will bind back to each other, or they can go into the full-length mRNA to give you an island, double-stranded, and so forth. So that creates a lot of impurities that you don't want. So the idea here, if you can control the abortive cycles by evolving the enzymes to control that, then you can eliminate quite a bit of those abortive sequences. So as you can see, the polymerase have a direct impact on the quality of mRNA produced, through abortive sequences, short transcripts, which lead to a lot of double-stranded RNA. So with that, the polymerase landscape currently is, as I just presented, the polymerase influence the efficiency and accuracy of mRNA synthesis. It is really important to control the abortive sequences during the IVT reaction. And the RNA polymerase quality truly affect all aspects of quality of the mRNA, from yield, fidelity, integrity, stability, and other aspects. So as Helgi mentioned, currently the most widely used RNA RNA polymerase is T7 or a variant of it. So T7 is known to have a high level of short transcripts, and that means the abortive sequences, which could lead to a high level of double-stranded RNA and other undesired impurities. It's not very efficient to synthesize long templates over 10 kb, and hence it impacts the self-amplified mRNA. And also does not perform very well if you're using native NTPs such as uridine instead of N-methyluridine. And also, as I mentioned, different T7 polymerases variants have been produced to address those issues, but really have not completely solved that. So the need is to have a polymerase that is diverse, that can work with various lengths of templates and sequences, can accommodate different capping strategies and capping analogs. It can also accommodate native and modified NTPs without any differences. And with that, again, I would like to emphasize that really there is no one RNA polymerase that could take care of all the needs for mRNA synthesis. So you might have to select the proper polymerase for your need. So with that, I'm going to be showing different case studies to show that not all polymerases are equal. And also, if you select the right polymerase for a long templates, you actually can get a much better results and performance than the regular T7. So the first study is, to show that not all polymerases are equal. For this, the experimental design was Primrose selected a linearized plasmid DNA of 5 kb. It includes the polymerase-specific promoter. So T7 have its own promoter sequence, and other polymerases will have their own promoter sequences that have to be taken into account, plus a native transcription starting site. Also, we incorporated 100 poly-A segment in the plasmid. The experiment was run using 3 different of DNA concentration. And this goes back to what Helgi pointed out, is we really want to reduce the input of material into the IVT reaction, that way so you can help with the cost. For this specific experiment, we did not use any capping analog, so those mRNAs were uncapped, and this was done on purpose so that we take out one variable out of the reaction. And we've used the worst case scenario is we're using uridine as a native NTP instead of N-methyl pseudouridine. The reason for that is it's widely known in the mRNA field that if you substitute N-methyl pseudouridine for uridine, just by doing that, you lower your double-stranded RNA. So in this case, Primrose, we decided to go with the worst case scenario just to see how we are controlling double-stranded. So the next is for the polymerase that were used in this study, we compared 5 different Prima polymerases, Prima 1, 2, 3, 4, and 5, as are named throughout the presentation here, versus a T7 RNA pol. And just to make a couple of notes about the Prima pols versus T7, the sequence between T7 and the polymerases are about 32 to 63% comparable. And also T7, the Prima RNA pols are not T7 variant. They are all unique and come from various genomes. Just keep that in mind. They are not T7 variants. And also the Prima pols that were used in this study, they were 1st or 2nd generation enzymes. And those are been taken currently to go through the 2nd and 3rd or 4th generation evolution to enhance them better. And this work is currently underway at Primrose. And so once all these were produced, then they were evaluated for yield, integrity, double-stranded, and sequence. In terms of yield, here we show this graph, the yield of all 5 prima poles versus T7 for the 3 different concentration of DNA template input at 2 nanomolar, 4 nanomolar, and the standard of 8 nanomolar that's currently used in most IVT conditions. As you can see, all 5 prima poles overall outperformed T7 in terms of the yield. Specifically, if you note that at the 2 millimolar concentration of input. Another thing that you can take from these graphs also is all 5 polymerases from Primrose also performed differently. Just to emphasize that point that not every polymerase is going to function across all the needs of mRNA synthesis. And what we've shown here also is the total mRNA yield, straight out of the reaction. So if you look at the target RNA, versus the DNA template ratio, which is the nanomole of the RNA target versus the nanomolar of the DNA template, at the low concentration of 2 nanomolar versus the standard concentration that's widely used at 8 nanomolar, you truly can see that all 5 prima poles have outperformed T7 in terms of true yield of mRNA. And in particular, if you look at prima 2 and prima 5, what are highlighted in blue, that is 320 over 300% increase in yield, which is, that by itself can reduce your cost quite a bit and streamline your process. Now, someone will ask as well, if I know I have a lot of total mRNA, but if it's not really clean, I have so much of it to clean, so at the end of the day, the yield is going to be the same. Well, if you look at the purity of all these mRNAs that were produced, again, the percent purity of these mRNAs produced by all 5 prima poles overall is outperformed T7. Again, I would focus on the 2 nanomolar concentration, where all 5 outperformed, gave a much better purity than T7. So you're not only getting an increase in yield, but also way much more of the mRNA that you need. And again, the purity varied between all 5 Primrose polymerases. And the reason for that is all designed for different purposes, and they will perform differently based on, for one sequence. So with that, then we looked at double-stranded RNA, which is something that everybody would ask when you start changing IVT conditions, specifically polymerases. So on the left-hand side, there is the graph that shows the comparison between 7 and all 5 prima polymerases. And you can see, as you compare all 5 of them to T7, there's at least 10 times reduction in double-stranded RNA. And here, the double-stranded is reported as nanogram of double-stranded RNA per mg of total mRNA. So once some people will see that size, so that's great, but I don't see the low level of the prima polymerases. So we took that and expanded the graph on the right-hand side to show the level of double-stranded that we are measuring. And you can see that worst case scenario, which is at 8 millimolar, one of the polymerases, it's still lower than 0.5 nanogram double-stranded per meg of total mRNA. And also, there is a variation that you can see between the input of the DNA template. At 8 nanomolar, overall, you see a higher level of double-stranded versus the 2 nanomolar, which is low input of the DNA template. And in addition, just to make the point again, that all 5 perform differently regarding the levels of double-stranded RNA. Yes, they perform differently, but still all 5 of them outperformed the T7 in terms of producing a much lower levels of double-stranded RNA. So from there, the question is, this is great. Can we find one polymerase that actually solve the problems for long template? Because as you know, going to self-amplified mRNA, there's issues with the percent yield that get produced, The double-stranded, it gets complicated because now the mRNA is much larger and you go through a lot of issues in terms of the purification, the downstream purification to get pure mRNA. So with that, we designed a case study to particularly target the long templates. So for that experimental design, we chose a linearized plasmid at 10 kb. Again, it includes the polymerase-specific promoter plus a native transcription starting site. And as the other study, we used 3 concentration of DNA concentration, no cap, same length of poly-A. But in this study, we actually looked at both native uridine and N-methyl pseudouridine. For this study, the RNA polymerases that were used were a little bit different. So we reversed it. We chose 3 different T7 RNA poles from 3 different vendors. And the IVT reaction condition was conducted per the vendor recommendation. So we want to do the IVT reaction for all these T7s at the optimum conditions that their vendor provided. And then we compared those to a Prima Extend. It's a polymerase Primrose RNA pole that Primrose designed specifically for large template over 10 kb. And for that, we used the Primrose IVT conditions that were developed with the Prima Extend. Again, just to make the point that the Prima Extend is unique and it's not a T7 variant. And in terms of product evaluation, we went through the same drill of yield of mRNA, integrity, and we also looked at purity by IPRP-HPLC and double-stranded by ELISA. As for the results, here these graphs shows the graph on the left is for the uridine and the graph on the right is for the N-methyl pseudouridine. As you can see from a yield standpoint, the Prima Extend way outperformed all 3 vendors for T7 polymerase, while achieving the same integrity in terms of the mRNA length. The same thing is observed for N-methyl pseudouridine, where also Prima Extend outperformed all 3 vendors for T7 and maintained the same length of mRNA. Then we looked at purity. So how does the purity look for this? And this is before the mRNA fully purified. It didn't go through any poly-DT or anything like that. I just want to make that point because you can enhance that purity with downstream. But here we just really looking at the IVT reaction to show that we eliminating most of the impurities before you even try to purify it. And this is what you need. We're eliminating byproduct at the synthesis part of the reaction. So again, the purity of Prima Extend outperformed T7 with UTP and with uridine and N-methyl pseudouridine. And this is this truly shows that this Prima Extend designed for a large mRNA of 10 kb or higher really is outperforming the current T7 from different vendors. Now, you asked the big question for double-stranded RNA. And this is where the huge illustration is of not all polymerases are the same. For double-stranded RNA, you can see the 3 vendors are all over the map in terms of the level of double-stranded RNA and the reaction. With that in mind, the Prima Extend outperformed all of them in terms of the low level of double-stranded RNA. Here we're showing the results from the UTP, but we also got similar results were observed with an N-methyl pseudouridine. So now this Prima Extend have shown that it works for long templates, 10 kbs and above, and all the indicators of the quality of the mRNA outperform the 3 vendors that we we tested randomly for T7. So this is a good choice if you are going to develop a long mRNA and you need the high quality and the purity. finally, we did a case study to understand, would this Prima extend? Is it compatible with all transcription starting sites? Is it compatible with all capping agents? Because if you're working on self-amplified mRNA, you're limited with your capping strategy to use CleanCap AU because it's targeted to start at the AT transcription site. So that way you can get the self-amplified mRNA. So, we said, let's look at that. And again, for this study, we used the same 10 kb as the case study 2 with the same polymerase specific promoter and transcription starting sites. We, again, the same conditions in terms of DNA template concentration. And in this study, we actually looked at mRNA capped and uncapped, and the capping was done co-transcriptional. And we've used uridine and N-methyl pseudouridine to cover the gamut of the experiment. As the RNA polymerases were used in this study, we selected only one T7 from a vendor, and we compared that to the Primrose Prima Extend. The IVT conditions for both were done under the same conditions, and for evaluation, we ran the same gamut of tests, and we added a little bit of sequencing data that we did to dive a little bit deep into not only the integrity of the mRNA, but also to understand those truncated sequences, what are they? And with that, the results here show 3 different transcription starting sites, a GG, AG, and an AT. And here you see a comparison of T7 versus Prima Extend for all 3 transcription starting sites. And as you can see, I'm not reporting here yield because of all the studies that we've shown, the yield actually always was higher for any Prima pol versus T7. But here we really wanted to focus on 2 things. One is the double-stranded RNA and the percent purity. And in terms of the percent purity, which is the bars, we outperformed T7 on every experiment. And also the double-stranded RNA was much lower than T7. And also the transcription starting site played a role in terms of the purity and the amount of double-stranded RNA, which makes a lot of sense that if you look back at the mechanism, this is where the stage where you're starting the abortive sequences. So it depends on, the transcription starting site does play a role into the mechanism of the polymerase. So here we are able to show that higher yield purity and lower double-stranded RNA with the Prima Extend as compared to T7 for 3 different TSS with and without CleanCap AG. CleanCap AG was used for GG and AG transcription starting sites, and CleanCap AU was matched with the AT transcription starting sites. So overall, from this experiment, that you can see that Prima Extend is compatible with various transcription starting sites and various cap analogs and capping strategies. Now, if you expand it a little bit more in terms of thought, you might be able to use AG as a co-transcriptional capping agent for a 10 kb or higher mRNA. So you really, over here, your capping strategy, you might, with this polymerase, you might be able to get away from AU and just use AG as an option. So during the study, we saw something interesting, and we wanted to dive a little bit more into it. And that is the purity of the mRNA. And again, when I'm talking about purity here, is straight from the IVT reaction with a simple workup. This did not go through any major purification to get ultra pure mRNA. And to the right is the chromatogram, an overlay chromatogram of the mRNA produced by PrimaPol in orange and the T7 in black. And to the right, the chromatogram is an expansion just to be able to see the little details of the impurity region. So as you can see, with the Prima Extend, you do not see a lot of impurities in that region, while there's a lot of, some of it in the T7, from the T7 reaction. And the question is, what are these? Are they really truncated, from the abortive sequence and so forth? So we were intrigued to see what those are. So those impurities were collected and they were analyzed by NGS to identify those peaks. And as you can see here, most of those peaks are identified to be between 0 to 1,000 bases. And this is where the Prima Extend actually helped the outperformance of T7 by controlling the abortive sequence and eliminating all these short mRNA that produce a lot of double-stranded and also will be difficult to purify, have a lot of it could cause a lot of work in terms of downstream purification of the mRNA. So from that, the question we ask is, does that have does the input of the template concentration have an impact on that? So we actually looked at that for the 2 nanomolar, 4 nanomolar, and 8 nanomolar concentration of DNA template concentration. And as you can see with 8 nanomolar, which makes sense, like why this is the standard, because you get the least amount of those abortive truncated mRNA from this IVT reaction. And they kind of increase when you go to 4 nanomolar or 2 nanomolar of DNA template concentration. And that's for T7. If you look for the Prima Extend, as you can see in the orange chromatogram there, it doesn't matter which concentration you use. Those truncated mRNAs are not formed. And this shows that the design and the methodology that Primrose is actually incorporating focus around controlling all these abortive sequences and the truncated mRNA that's produced so that it can produce a very high quality mRNA. And as you can see from the graph below, it kind of explains also the level of the double-stranded mRNA in the reaction. So as you would expect, the least amount of double-stranded you would see is with the 8 nanomolar. And the 4 and the 2 give you, because there's more truncated mRNA, it gives you more double-stranded in your reaction. So in summary, a Prima RNA pulse outperformed T7 at a standard and a low DNA template concentration for yield, integrity, and double-stranded RNA. And also we showed that not all RNA poles are equal, and each of the 5 Prima RNA poles that were tested had a different performance than each other. For long template, Prima Extends outperformed T7 actually from multiple vendors on yield, integrity, purity, and double-stranded. In addition, we showed that Prima Extend is compatible with various transcription starting sites and is compatible with different capping analogs and capping strategies. Now, with that, I'd like to turn it back to Helgi to conclude this webinar.
Thank you, Khaled. Yeah, so we'd like to wrap this up by showing you a little more about Primrose's mRNA offerings, mRNA manufacturing offerings. And as you've already heard, what we bring to the industry is centered around our RNA polymerases. That's where we began, and that will continue to be our focus. And this is now an offering of RNA polymerases for sale. At the bottom left, you can find one. Prima Extend is for sale on our website. And then this is coupled with very active RNA polymerase improvement programs that we either run in-house, and we're constantly producing higher performing enzymes, or performed together with a partner to meet their specific needs. In addition to that, the panel of RNA polymerases that we have in-house, we offer for evaluation, and that on the far left. So that evaluation service and offering has been in place for several years and has been actually a very successful route to give a potential customer a sense of the performance of our enzymes compared to the standard. Now, in addition to these offerings centered around RNA polymerases, we also offer mRNA analytical solutions. As you can imagine, running all these reactions and characterizing them means that we have to develop the ability to fractionate mRNA and test its quality. These are highly developed assays at Primrose, and we're happy to engage with a company that says we know how to make our own mRNA, but we don't fully know how to characterize it. So very happy to work with customers like that. And then moving towards the right, IVT process development is something that we also offer. And finally, RNA polymer manufacturing, of course, linked to the RNA polymerases we're already selling, and also linked to the custom improved RNA polymerases that we develop in collaboration with specific partners. I wanted to also come back to the value proposition of why switch RNA polymerases, why spend the time and the trouble to use our reagents compared to T7, which I believe in the hands of most people at first glance is a perfectly adequate enzyme. And the reason is summarized in this value proposition diagram is we offer lower cost, higher efficiency, and lower cost. Now, the cost variables relate to yield, increased yield, reduced double-stranded RNA, and higher yield of the target mRNA that's linked to the higher purity that Khaled covered really in great detail in his slides, so leading to reduced purification costs. Those are the primary RNA polymerase performance indicators that affect cost and lower it. The secondary ones, as we evolve our polymerases to higher efficiency, are linked to further increases in yield and purity, but then also to higher incorporation efficiency of the cap analogs. And this is where the ability to use lower concentrations of cap analog at equivalent capping efficiency can then greatly reduce the cost further. And finally, and again, really well illustrated in the data that Khaled showed, utilization of the DNA template at high efficiency, so you get the maximal number of RNA molecules out of each molecule of DNA template. And this is an area where we will continue to work and continue to make improvements, because in principle, there's no reason why it has to cap at 1,000, which is where we're now. It could go much higher than that. Finally, I'd like to give a shout out to our Extend kit that's for sale on our website. So please take a look. Please buy it if you're interested. Please contact us if you have questions. This polymerase is aimed towards long template mRNAs. We believe this can become the go-to RNA polymerase for long mRNAs, produces high quality RNA that is long above 10 kb, and it's available in R&D and GMP grades. And finally, I'd like to acknowledge the team. We have, this is a portion of the company, about half of the company shown here, and between molecular biology, enzyme evolution, and synthetic biology, analytics, RNA and protein analytics, and protein production, you can imagine that this is a complex effort. It's a complex effort of highly trained scientists working hard, and these efforts continue and are constantly giving new polymerases that we hope will enable the fantastic new mRNA-based drugs and vaccines that are currently under development. And so it's good to close with giving you a sense of our mission. Our mission is to help you, drug developers, vaccine developers, do your work better, and to contribute our advanced reagents to your drug and vaccine development.
Well, thank you, Helgi and Khaled, for a great presentation. Now we're going to start our Q&A, so please join me on camera, and I hope you're ready for some questions. And to our audience, please do keep your questions coming in. We'll get to as many as we can today, and we'll answer any others by email. And if you would like me to direct your question to a specific presenter, you can indicate that in your message. Okay, so first up, I have quite a long question in from the audience, so please do bear with me. The vibriophage RNA polymerase in the new Extend kit still uses GGG as the initiation nucleosides, since this is targeted towards longer mRNA transcripts of self-amplifying type. So will it be suitable with templates that include the AU initiation required for the use of CleanCap technology?
Yeah, I can take that. Yeah, so I think Khaled addressed that question clearly with his slide that showed the utilization of the 3 different transcription start sites by PrimaExtend, GG, AG, and AU. They're both, all 3 are essentially equivalently utilized. And that translates into efficient use of the caps. We didn't show capping efficiency here, partly because that characterization is still underway, and partly also because we're constantly working on improving that. But to answer the question clearly, yes, absolutely. This polymerase can handle all 3 different transcription start sites.
Fantastic, thank you. And next up, Helge, another question here for you. Do you see pronounced performance differences between RNA polymerases as it relates to the size of mRNA?
We do when it comes to a specific polymerase. And among the 5 primas that Khaled presented early in the first case study, there certainly is a difference, as there is with T7, in efficiency and yield as you go to higher template length. However, PrimaExtend is really good at long templates, and it's also good at short templates. It's a good all-round enzyme. So while the template length dependence is something of concern for general polymerases, PrimaExtend is specifically designed to take care of all lengths.
Fantastic, thank you. Next audience question here. How was the protein expression and innate immune response from these mRNAs in mammalian cells?
Yeah, so we can only comment on this very indirectly because most of this work, this type of work, has been done in collaboration with partners, and this is not data we can present or comment on directly. However, the RNA quality that we see in vitro translates into excellent translatability. In fact, that was one of the first characteristics we tested when we first developed our panel. We were focused on generating RNA with higher translatability. So you can generally assume that higher quality in vitro means higher translatability. However, this is also work that's underway in our hands, and we hope to have data later this year where we can show that in vivo, in cultured cells. But we're not a drug developer, so we're very happy to work with you in your tissues, in your target organs, or animal models to show that our mRNAs produced with our RNA polymerases are very, very efficient.
Fantastic, great answer, thank you. And next audience question here. Is Primrose Pol planned to be GMP ready soon?
Yes, so we are ISO certified. Our facility is ISO certified, and that means we supply polymerase for a GMP mRNA manufacturing process. That really covers it. We're happy to discuss details with you.
Fantastic, thank you. And next up, so what would you say to a company that is currently using T7 around how they would benefit by switching to Prima RNA Pol Extend?
Well, the data shows that the performance overall is outperforming T7, but also Helge have shown in terms of the reduction of cost and also the simplicity in terms of the downstream process. So those are the benefits that you would get. But in order to switch, you would have to do some comparables depending on where your drug development stage is. If you're in the early stages, you can switch. If you, let's say, finished phase 1, you would have to do a comparability study to show that introducing a PrimaExtend versus T7 it's really not impacting the therapy indication at all in terms of translation and so forth. But you'll get a lot of benefits in terms of cleaner mRNA, easier downstream process, and a huge cost saving once it's all applied together.
Fantastic, thank you. And another audience question that's come in here for you. How much loop-back double-stranded RNA is present after IVT reaction?
That's a very good question. And also it's very difficult to answer, because one of the pitfalls of measuring double-stranded RNA is we really don't understand what type of double-stranded RNA are we truly measuring. Is it islands? Is it full length? Or is it a loop-back? Now, loop-backs could be disrupted, by adding, by sample preparation to see if it's actually a loop-back or a true double-stranded RNA. We did not distinguish between those. We were looking overall in terms of reduction of a total double-stranded RNA, regardless if it's a loop-back or a full length or an island.
Fantastic, thank you. And next up, let me see, an audience question that's come in here. Did you check the fidelity of these enzymes, and are they different from T7?
Yeah, I can take that one. It's an excellent question. It's something that we're actively focused on. We've done an initial fidelity comparison by next-gen sequencing, and we basically didn't observe any changes. However, very likely most of the errors observed in that analysis are sequencing errors, not RNA polymerase errors. So there's a huge literature around this, as you undoubtedly know. We're trying to develop very specific, accurate, dedicated fidelity assays to use on the polymerases, and we should have that data characterizing our primas and characterizing prima extent later this year. But I think for now, it's safe to say it's highly unlikely that these polymerases have error rate much different from T7. And I think T7 generally is recognized to be sort of a well-performing polymerase when it comes to error. Not great, not great. There's room for improvement. And this is probably an area where we will direct additional efforts in the future.
Absolutely. So watch this space. Another question that's come in from the audience here. In addition to Extend, are the other Prima Pols 1 to 5 also available commercially?
They are not available commercially, but they are available for evaluation. Please contact Polymerase and we'll be more than happy to work with you on that.
Absolutely. So be in touch to that audience member. Next up, impressive data. This person says, apart from the differences seen with different lengths of RNA, do you see differences with different sequences of the same length?
Yeah, that's a really good question. Yes, we do. But a polymerase that's good at one long length tends to be good at another long length. So we've compared, we have a human cDNA that we use internally, 10.7 kb in length. We routinely compare that to the standard VIV self-amplifying RNA sequence, and polymerases that do well on one do well on the other.
Thank you. Now, I think we have time to squeeze in maybe just one more question. Do you require to change the promoter every time you change one of your enzymes?
So our enzymes, so the 5 primas and prima extend, have distinct promoters, and they're different from the T7 promoter. So if you want to test them, you would have to incorporate those promoters into your constructs, and we're happy to help with that. However, one of our planned releases later in the year is what we call a no-compromise enzyme. No compromise meaning all-around high performance in terms of capping, yield, integrity, and ability to transcribe templates of different sequence and length. And our goal there is to have probably 2 enzymes released, one that is a truly excellent enzyme, and one that is probably just a little bit lower in performance, but that recognizes the T7 promoter. So stay tuned, and we will be able to, if you're unable to work with other promoters, and again, we can help there, But if you really are married to the T7 promoter, we will have offerings related to a T7 promoter compatible polymerase later in the year.
Yep, absolutely. Thank you. That's a nice note to end on. So thank you, Helgi and Khaled, for answering those questions. I'm afraid that is all we've got time for today. But any questions we didn't get to, we will reply by email. I'm aware there was quite a number of audience questions still coming in. This webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar. The link to this can be found in the resources on the right-hand side of the screen or in the on-demand email. And additionally, an attendance certificate is available on the right-hand side too. So all that's left is to thank our speakers once more for a wonderful presentation, and thank you to the audience for listening. We hope you'll join us again soon.
Thank you for joining. Great pleasure.
Thank you.